Okay, the purpose of the pentose phosphate pathway is to generate NADPH. Now recall we have seen a lot about NADH. NADH is an electron carrier that is ultimately going to be used to generate energy. NADPH, although only a small modification to the NADH structural backbone, this is providing reducing power for biosynthetic reactions. NADPH and NADH are not interchangeable in these functions. Now, in order to produce this reducing power for biosynthetic reactions, we are of course going to be in a fed state. The organ that is primarily going to be using this pathway at the greatest extent, although all cells can do this, is going to be the liver. So in the fed state in the liver, we have glucose coming into our cell. Our first step of our reaction is glucose 6-phosphate by the hexokinase enzyme. And this will trap the glucose inside the cell. Then we have our isomerization reaction to produce fructose 6-phosphate by the phosphoglucose isomerase. And then, of course, our gatekeeper enzyme, PFK1, to generate fructose 1,6 bisphosphate using ATP. Now our gatekeeper here again of PFK1, this gatekeeper is the gatekeeper to glycolysis. So as the energy charge in the cell increases, we are going to inhibit the PFK1 with increasing concentrations of ATP. So this is the same type of activity that we saw in glycogen synthesis. Being in a fed state, we can afford to expend energy for making biosynthetic molecules rather than energy. So if we block PFK1 because our energy charge is full, then our fructose 6-phosphate increases. Recall that the isomerase reaction is reversible if this sub becomes highly concentrated substrate and product here. This becomes a negative delta G. Free energy is released in this direction, so we reverse our glycolytic pathway by this step, but then also recall that the hexokinase reaction is not reversible. So similar to glycogenesis, synthesis of glycogen, we are building up our glucose 6-phosphate. Now at this point we can enter either glycogen synthesis or the pentose phosphate pathway. So the first step in the pentose phosphate pathway is mediated by glucose 6-phosphate dehydrogenase. Being a dehydrogenase, this is a redox reaction. So this is going to be our first step of generating our first NADPH. We generate, the product is 6-phosphogluconolactone. This is part of the intermediates of the pentose phosphate pathway that are not exceptionally important. The next step here is lactonase, and we are going to form 6-phosphogluconate. Now at this point, we are going to be generating our second and final NADPH. The enzyme for this reaction is 6-phosphogluconate. gluconate dehydrogenase. And the product of this reaction is ribulose 5-phosphate. Now, also important to note here that this is a glucose-based molecule, so this is a 6-carbon molecule. Ribose is a 5-carbon molecule. So that means the second step in the pentose phosphate pathway to generate NADPH is an oxidative decarboxylation reaction. So we are losing CO2, losing a carbon at this point. Now the ribulose 5-phosphate is a ketose because it has a ketone in it. This can undergo change to xylulose, phosphate, or we can isomerize to ribose 5-phosphate. Once we have ribose 5-phosphate, this now becomes our precursor for nucleotide synthesis. So in order for the ribose 5-phosphate to enter the nucleotide synthesis pathway, this has to be converted into phosphoribosyl pyrophosphate. It's abbreviated PRPP. This particular step is inhibited by purine nucleotides, specifically ADP and GDP. So this regulation simply indicates that when we have sufficient amounts of purine nucleotides, it is going to feed back and inhibit on nucleotide synthesis at the very first step of preparing the ribose structure. What this allows us to do is if we have sufficient nucleotides, we can take the ribose together with the xylulose 5-phosphate, and we can perform a series of reactions that will return these rich carbon sources back to glycolysis. And they return the products back to glycolysis at the level of fructose 6-phosphate and glyceraldehyde 3-phosphate. There's a series of reactions that occur in here for this transformation, and the primary information is to know that the enzymes are called transaldylases and transketolases. Now, as far as regulation of this system, the first step of the pentose phosphate pathway with glucose 6-phosphate dehydrogenase is the rate-limiting step and the entrance into the pathway. The activity of this enzyme is going to depend on the concentration of NADPH. So if NADPH is significantly high, we have sufficient amounts, this enzyme, the dehydrogenase, will be inhibited. In that case, the glucose 6-phosphate is not able to enter the pentose phosphate pathway. If NADPH levels go down because of biosynthetic reactions, then the glucose 6-phosphate in the fed state can enter the pathway to generate more. So under conditions of high NADPH, where the pathway is inhibited, the glucose 6-phosphate can only go in two directions in a fed state in the liver. It can either go into glycogen synthesis, or it can continue down the glycolytic pathway if the gatekeeper allows it.